Do you see the slides?
Yes, that's all perfect.
Okay, very good. So yeah, thank you very much for having me and for the kind introduction. My name is Amelie Luoma. I'm a researcher from IRIS Group coming from Finland in Turku. And I hope that I can stimulate some synapses today by sharing our research about synaptic density in multiple sclerosis. And as you said, this is an in vivo study, so a study in the living human brain, and it uses PET imaging, so positron emission tomography. There we go. So first of all, I would like to say a few words about multiple sclerosis, or MS in short, as not everyone might be so familiar with it. So MS is a neuroimmunological disease, and unfortunately, it can affect already relatively young patients, young adults. The average onset is around 20 to 40 years, and it can then cause physical and cognitive disability. And the pathogenesis of MS is rather complex and today still not sufficiently understood. But what we know is that there are, on the one hand side, focal inflammatory events, and on the other hand, there are chronic smoldering processes which already appear at the early stages of the disease and then become more prominent in later stages of the disease. And this also a little bit reflects in the typical appearance or phenotype of the disease. And while there's a great heterogeneity, and one can really say that every MS patient has its individual MS course, one can see relatively often in the beginning relapsing and remitting phases. So neurological disability appears and then they go into remission again, so it disappears again. And then after some time, the disability starts to accumulate, and after some years, quite often the disease turns to a progressive phase, so where then there is no relapses anymore visible, and we just have slow progression of the disability. And when we see a patient in the clinic, the gold standard in the clinics is still an MRI. So each patient gets an MRI. And in the MRI, we can typically see lesions in the brain, CNS lesions, and also in the spinal cord. And most prominent, they are in the white matter. So they are practically scar tissue. That's also why it's called sclerotic, so multiple sclerosis. And on the other hand, we can also see neurodegeneration, so atrophy. And while white matter pathology is a little bit easier to see, especially gray matter pathology, what is important for here, because there's also the synapses located, is harder to see in the MRI. And especially also smoldering processes, which are much more subtle than these acute inflammatory events, are difficult to see and to visualize in the MRI. And that's why especially PET imaging starts to be valuable to detect the pathology behind MS. In fact, the topic synaptic density in MS is relatively unexplored. That means that everything that we know about MS, synaptic loss in MS today, is still based on histological postmortem studies. So brain examinations after people have died and they have donated their brains. And indeed, those studies, they could show that there is synaptic loss in the gray matter, as well in the lesional gray matter, as also in the normal appearing gray matter. And one of the milestones in these histopathological study examinations was a paper by Jürgens et al. And what they did is they practically visualized dendritic spines with Gold-Seacock staining. And here you can see they looked at controls at normal appearing gray matter and at lesional gray matter, and as well as in lesional as also in normal appearing gray matter, they found a reduction of the dendritic spines. That is where the synapses are located. However, what is important for this study now, we don't have any reference in terms of in vivo studies to compare this study with. So what we did is we did PET imaging, and we used a carbon band tracer called 11C UCB-J. And this tracer binds to a presynaptic vesicle protein, which you can see here. So it's in the presynaptic area of chemical synapses. So we look only at chemical synapses here. And this isoform SV2A especially is very valuable to quantify synapses because it is ubiquitously presented throughout the whole entire adult CNS. And this method, UCB-J PET imaging, has shown to be valuable already for other diseases or for the investigation of other diseases. Just to name some, in Alzheimer's, schizophrenia, or Parkinson's, they could already visualize synaptic loss with this tracer. So at the core of our study was therefore the PET imaging, which you can see here. And this involved 10 MS patients and 8 healthy controls. And at the same time of the PET imaging, everyone received an MRI as an anatomical reference. Additionally, all MS patients underwent clinical and cognitive evaluation. So we did this in MS with a typical score, EDSS, which is typically used in the clinic also for MS patients. And additionally, we did a cognition test with SDMT, which is primarily a measure of the information processing speed. Additionally to that, for the MS patients, we included in this study data that reached 5 years back, so 5 years prior to PET imaging. And for the MS patients, we had here an MRI and also clinical and cognitive assessments available. So here's the demographic table. This is a bit of an overkill, but I will walk you through this. So on the left-hand side, you can see the demographic data at the time of the PET imaging. So 8 healthy controls and 10 MS patients. And you can see that the two groups in terms of sex and age were well matched. And you can also see that the average age of MS patients was about 53 years. EDSS was a little bit higher than 3, and the cognitive test SDMT was around 50. What you can see here in the lower part is that in terms of volumetric measures like brain volume and cortical gray matter volume, there were no differences between the two groups. Then we looked at the change that is relevant for the MS patients, so what had changed during the 5 years prior to PET imaging. And we can see that the EDSS, so the clinical measure, patients had gotten a little bit worse, on average 0.5 points, and they had gotten a little bit slower in the information processing speed. And importantly, they also had significantly lost some cortical gray matter volume. Okay, so after a lot of processing and reconstruction, we eventually ended up with these beautiful pictures here. So what you can see here is in the background, we have the MRI, and on top of that, we have the PET image. So the colorful one is the PET image. And in fact, you don't see here individual subjects or images of individual subjects, but what we see here is the mean images of the two groups. So in the upper row, we have the mean image of the healthy control group, and in the lower row, we have the multiple sclerosis mean images. And the PET image here represents VT volume, VT, which is the volume of distribution, a measure of the synaptic density. And the higher the number goes, the more synapses we assume in that area. So if we talk in the terms of the color code, we can say that the more red or yellowish we see here in these areas, the more synapses there are. And when we now compare these two rows, we can clearly say that the healthy control group seems to have more synapses than the MS group. So as a next step, we defined areas that we wanted to closely investigate. And we defined the cortical gray matter as primary region of interest. And here you can see we defined this with free server, and the free server generates masks that we can then use. And then we subdivided the cortical gray matter, well, practically into the different cortices, so cingulate, occipital, temporal, frontal, and also some insular cortices. And additionally, because this is the first time that the synaptic density in MS is explored in the entire brain at once in this in vivo imaging, we defined some exploratory regions to see what goes on in the other areas of the brain. And therefore, we used amygdala, caudate nucleus, cerebellum, hippocampus, putamen, and thalamus, without then further subdividing those areas. And indeed, when we then look at the actual numbers, the results that we already saw in the PET images, they got confirmed. So we really have a significant difference here. In the first box plot, you can see the cortical gray matter, and here this is the VT, so the synaptic density, and white is the healthy controls, and this orange yellow one is the MS. So we can see that they are clearly lower. And when we look at the different cortices here on the right-hand side, in which we subdivided the cortical gray matter, we see that all of the areas support our main finding in the cortical gray matter. Also, we can nicely see that in the white matter, there's much less synapses, and we also don't have a difference here between the two groups. Okay, of course, we were then also interested whether we would see any correlations between the synaptic density and the clinical and the cognitive measures that we had assessed during our study. And while there was no correlation between the clinical measures, so the EDSS, and the synaptic density, we could see that there's a significant positive correlation between the cortical gray matter VT and the SDMT. So in other words, the faster someone performed in the information processing speed test, the more synapses he also tended to have. And interestingly, we then also found a significant correlation when we looked at the SDMT change and the cortical gray matter VT. That means the more someone, or the worse someone got during the 5 years prior to PET imaging and the PET imaging in that test result, the less synapses also he or she tended to have. Then we also looked at the exploratory regions, which you can see here in the upper box. So on the left-hand side, you have the different regions. And then here, the second column and the third column are the VT values, so the synaptic density synaptic density measures. And in the end, you have the p-values. And we can here see that the amygdala, putamen, and thalamus, they had significant differences between the two groups, while the other ones, caudate, nucleus, cerebellum, and hippocampus, did not show any significant differences. And we then also were curious whether we would see a correlation in those areas with the cognitive test results that we had. And indeed, in most of the exploratory regions, we also saw a correlation between the synaptic density and the information processing speed, as you can see here in the lower box. And when we then looked at the SDMT change during the 5 years prior to PET imaging, we see that even more regions were involved. And also putamen and thalamus became significant here on this level. Okay, I would like to come to the take-home message. I would like you to remember that the synaptic density in multiple sclerosis can be indeed quantified using 11C UCB-J PET imaging. And with this in vivo study, we for the first time show synaptic loss in several brain regions, including cortical regions, amygdala, putamen, and thalamus, and thereby we complement histopathological studies, which have shown that already previously. Furthermore, the synaptic density in MS seems to be of great importance when it comes to cognitive measures. Last but not least, I would like to thank my wonderful research group in Finland and the financial supporters of this study. And I would like to share my disclosures with you. Thank you very much for your attention.